Well, I'm happy to introduce the next speaker, who's Dr. Gerald Meininger from the University of Missouri. He's going to speak on vascular smooth muscle mechanotransduction, a tug-of-war.
My thanks to the organizers and the chairs. I appreciate the opportunity to speak before you all. Today I wanted to focus on vascular smooth muscle cells and try to expand a little bit about what mechanotransduction is to me. I want to provide a little bit more of an explanation about what mechanotransduction is, and that I think it's part of a system of responding to mechanical forces that involves, importantly, mechanosensation as well as mechanotransmission. And this mechanotransduction process is basically just a way of translating mechanical forces that moves in a two-way street. It's clear that this process of sensation, transduction, and transmission plays multiple roles in the circulation. For example, it's very important for regulating contractile tone. We know that the regulation of contractile tone is linked to blood flow regulation. We also know it's linked to vascular wall and cellular remodeling and development, for that matter. And we know that it's linked to many different disease processes. And just a few examples here: hypertension, diabetes, and even large vessel stiffening, even though this is a small vessel conference. In today's talk, what I want to do is just briefly introduce some background and traditional concepts, and then try to stress the fact that the vascular wall responses to mechanical force and the cellular responses to mechanical forces are very dynamic and adaptive in nature. And then finally, where do we go from here and some possible future directions. Now, just as a very quick review of what the major mechanical forces are that we're dealing with in the vascular wall, there's shear stress, which impacts primarily the endothelial layer or intima. There is circumferential tension or stress, which is introduced by the intravascular pressure in the wall. And there is longitudinal stress, which is not frequently talked about, but it is a stress that we find in many of our peripheral vessels. The other thing I wanted just to ask you to look at here is when you look at the adventitia, media, and intima, these diagrams we often see in the textbooks make it look like the vessel is a layered cake, that each layer is totally independent from one another. Our smooth muscle cells are nice and uniform in shape and wrap circumferentially around the wall, and I'll come back to that point. Now, when we think about how vascular smooth muscle cells sense, respond, and transmit force, one of the first things we are driven to think about a little bit more is, well, how are they connected? And how is that force getting into cells? And so this is just a brief summary of some of those major routes. There are vascular smooth muscle cell to matrix connections, which are governed primarily by a family of proteins called integrins. There are vascular smooth muscle cell to smooth muscle cell junctions, adherens junctions. In smooth muscle, this is primarily N-cadherin. And then we have endothelial cell to smooth muscle cell junctions, which occur in the fenestra of the internal elastic lamina between the endothelial layer and smooth muscle cell layer. And finally, a little studied area is we also have smooth muscle, and I'm kind of generically grouping a whole bunch of cells together to mural cell connections. And these could include neurons, they could include pericytes, they could include fibroblasts. And this is a little studied area. Now, one of the most widely used preparations in this field has been the isolated perfused arterial. It has a number of strengths which have allowed many advances in the field of looking at mechanotransduction. These strengths include we can control the mechanical forces. We can control the intravascular pressure. We can control the flow through these vessels. We can fluorescently label different components of these blood vessels. And we can also label proteins and look at their spatial distribution. We can use conventional light microscopy on these vessels, and we can use more advanced fluorescent microscopy techniques like confocal and two-photon microscopy. And lastly, these vessels can be biochemically assessed if we're interested in cell signaling pathways. Weaknesses of this technique: it involves removing the vessel from its native environment. So we are studying it in isolation, but that's also a bit of a strength for the technique as well. Just a little bit of history here in terms of the origins of mechanotransduction in the vasculature. This probably dates back to Sir William Bayliss, who was the first to publish a paper indicating that the vasculature is responsive to changes in intravascular pressure. And this is usually diagrammed in single arterioles with a pressure diameter curve that looks like this, where if the vessel is passive with no tone and you increase pressure, it distends along a passive distension curve. If the vessel has tone, and in a normal vessel, this is about 100 micron arteriole, the pressure is going to be between 80 and 90 centimeters of water. As you increase pressure, the vessel will contract. As you decrease pressure, the vessel will dilate. And I also want to point out here that this same relationship exists with or without the endothelium present, indicating that it is intrinsic to the vascular smooth muscle. That's not to say that endothelial cells cannot contribute, but the basic response to the pressure and force is the smooth muscle cell. Now, we wanted to look at the cells within the wall, so we developed an imaging approach that allowed us to look at individual smooth muscle cells. And this shows an arteriole that would be running this way across the wall. And we used a technique called fluorescent dye exclusion, where we simply added fluorescein to the vessel bath. The fluorescein leaks in and around and between the cells, and then we can invert that image to get a nice picture of the smooth muscle cells. And what I want to point out is if you look at these cells, they're not nice and uniform and all oriented in the same angles and directions. They have different shapes. They have different relationships with one another. It really looks like a heterogeneous population of cells, and that's what I believe it probably is. Now, we did a very simple experiment to look at how these cells transmit force and how they sustain that transmission of force. And we did that by adding norepinephrine to one of these vessels and following the shape of the cells. And this next slide just summarizes that experiment very briefly. When you contract an arteriole, and in this case it was using noradrenaline, and we looked at the vessel 5 minutes after we took a through focus image series, and look at the individual cells, and we've pseudo-colored 2 cells here just to look at for a moment. The diameter was 91 microns. The overlap between these 2 cells is about 16 microns. When the vessel contracts, you can see the cells shorten, you can see them fatten, just the way we conceptually would envision this process. However, if you leave the noradrenaline on this vessel for 4 hours and look at those same 2 cells, what we noticed is that the cells are reestablishing their initial length. The overlap between them has increased. They're moving past one another, yet they're still constricted. And we have this change in overlap. So the cells are not cemented in place. They can rapidly adapt, change position, either by changing relationship with the matrix, changing relationship with each other, or cytoskeletal remodeling. And I believe probably all 3 of those are taking place. This next little movie I wanted to show is just to illustrate and attack this layered concept a little bit. In this particular image, the smooth muscle cells are going to be labeled green, as I showed in the previous image. The red is elastin fibers in the vascular wall. And I just want to show how complex this network of elastin is and how it changes as you penetrate through the vascular wall. And what is apparent when you play this over and over and over again and study it is that if you look at the outer layer, most of those elastin fibers run in the longitudinal direction. If you snip them, the vessels lengthen, so they do keep the vessel under a longitudinal compression. The medial layers run mostly circumferential, not longitudinal, and there are penetrating perpendicular fibers that pass all the way down to the internal elastic lamina and attach to it. So it's an integrated structure that runs all the way through the wall. And this structure changes depending upon where you obtain your vessel, what stage of development you're in, and it's quite different when you look at cerebral vessels. I feel I had to bring that up. The cerebral vessels totally lack the external adventitial layer, and all you see is the internal elastic lamina. So they're very sensitive to longitudinal stress. So as a brief summary, not all vascular smooth muscle cells are the same. Cells are dynamic. They have a positional plasticity. The ECM is an integrated functional part of the vessel wall and needs to be understood just as much as we understand the living part, the cellular part of the wall. If the matrix is disturbed, function is disturbed. And we postulate that this dynamic process is an efficient means of tuning the contractile state of the smooth muscle cell to the mechanical state that exists in the vessel wall and the mechanical environment. Now, we took this to the cellular level. To do this, we used an atomic force microscope where we basically, at the heart of the system is a cantilever with a tip that we can interact with cells. We isolated our cells from microvessels. We can position this tip and we can record the forces that are encountered at the tip. And we can calculate things like the cell stiffness or elasticity as well as adhesion force. This next little movie is one example of how we do this, where we take an AFM probe, we coat it with a matrix protein. It might be fibronectin or collagen. We indent the cell. You can see the deflection. We use this information to calculate the estimate of elasticity using the Hertz model. When we retract the probe, if there are adhesions between the fibronectin coating the probe and the cell, we get a slightly different retrace curve. We can use this information to calculate adhesion or how sticky the cell is. And we did a very simple experiment. We treated these cells with angiotensin II or adenosine, and we looked at stiffness. And this column of graphs right here shows a single cell example. This shows a group example. What I want to point out is if you look at the angiotensin II example, this bit of black here is the control period. A2 was added at the dashed line. And I want you to notice how quickly the stiffness starts to rise in the cell. And I also want to point out that this is cortical stiffness, since we're only indenting the cells about 150 to a maximum of 300 nanometers. So it's just the very surface of the cell. Also interesting, I don't have time to go into it today, angiotensin II throws the cells into an oscillation pattern that you can record. And that's what you see happening here. And with adenosine, we saw just the opposite. We see a decrease in the cortical stiffness of the cell. You can also assess the adhesion of these cells, and we did this by looking at that retraction curve. And what we notice when we look at adhesion, if you look after angiotensin II, there's a marked increase in the density of adhesion events. There's many more adhesions between the probe and the cell, and when they break with higher force. If you look at adenosine, you see just the opposite. We see a decreased density of adhesion events, and the breaking force is declined. So both of these things indicate that when cells contract, they get stickier. When they relax, they get less sticky. We also wanted to look at remodeling of the cytoskeleton. We did this by using the AFM in a scanning mode where we get cell topography, just scan back and forth horizontally. We can reconstruct a height image of a single cell. This is a single isolated smooth muscle cell. And this is a height map, so the brighter area is high. You can pseudo-color it and see that this cell rises about 4 microns above the substrate over the nucleus. And we can also process these data so we get a deflection image, which allows us to delineate the stress fibers underlying the cell surface membrane. Now, just a real quick analysis scheme that we used. This shows pre-angiotensin. We scanned in high resolution a 40 by 40 micron square, shown in blue. And we drew a line across this cell and looked at the scan after angiotensin 2. There's an increased number of stress fibers. The stress fibers are bigger, and if you do an analysis of orientation, they show that the orientation is increased. Whereas angiotensin 2, we saw just the opposite effect occur. In fact, the angiotensin, not angiotensin 2, just adenosine seems to cause a depolymerization of the cytoskeleton and disorients it. And nitric oxide has even a more profound effect. So in summary, smooth muscle cells are very mechanoadaptive in the wall. The adaptation involves rapid changes in the stiffness, stickiness, and cytoskeletal structure of the cell. And it supports the conclusion that the cells themselves tune themselves to their mechanical environment and the mechanical work that's placed upon them. Lessons learned: mechanotransduction is multifactorial. There's more than one sensory pathway and molecule. There are many signaling pathways that are involved, which I didn't go into. And the process can be modulated by multiple cell types, circulating factors, and cytokines. The process is dynamic. It involves more than changes in contractile tone. It's more than just actin-myosin interaction. And it operates on a temporal continuum, so that anytime you look at a model system and perturb it mechanically, you're looking at a snapshot in time, and it's going to change depending on when you look at that. And then gap analysis: what do we need to improve our basic understanding of mechanotransduction so that we can use this information to fight disease? We need more knowledge of the time-dependent events. We need a much more detailed understanding of the extracellular matrix structure and where cells interact with it and how that structure behaves. We need to look at intracellular interactions. We need to look at how integrins interact with cadherins and how one cell interacts with another. We need more detailed understanding of the cytoskeletal compartments within the cell and how they behave differently. And finally, technically, we need much better visualization approaches, faster ones. We always want more resolution, more speed. That's common for biologists to want that. And I think, in addition, and I heard this earlier, we need models. We have spent a generation breaking the system down into smaller and smaller parts, and it's time to start reassembling this and working as teams, mathematicians and biologists, and building models that are predictive, that can be tested, so we can go back and forth. And I think this will improve the speed with which new concepts develop. Thank you.
Thank you. I think we have time for just one question. Well.
Yeah. Thank you, Jerry. At INSCO, University of Alabama, Birmingham. You sort of did a nice job of depicting what the intricacies of this mechanosensation process is like, but what happens to this process under conditions of chronic hypertension or even acute changes, of marked changes in pulse pressure?
Those are good questions. We're looking at hypertensive cells now. They are different from normotensive cells. They're much stiffer, they're much stickier. We haven't yet done any dynamic studies to see how their responses are different.
Jerry, I'm going to ask just a very quick one. The endothelium is critically important in the cerebral vasculature, as you pointed out. You're denuded endothelium, yet you still got smooth muscle effects. What if you damage that endothelium? Hypertension, rheologic factors, tortuosity factors, anything that would affect that endothelium. Then what's going to happen to your smooth muscle reactivity?
short answer is we never did those studies. It's a good idea, but we haven't done that.
Thanks, Jerry.
So,